Okay, so number 3 of our shorter questions, these are typically, I'd say, like 4 to 6 marks. This one says to investigate whether red blood cells of animals lose the ability to take in glucose from their environment as they age. Scientists collected red blood cells from guinea pigs that ranged in age from 1 day old to 7 months old. Scientists incubated an equal number of red blood cells in separate culture dishes that contained a 300 nanomolar solution of radioactively labeled glucose. The amount of radioactively labeled glucose present inside the red blood cells of each group was measured over time. Okay, so A is a pretty straightforward question. It's just asking you to describe a difference between passive and active transport. Now, it's asking you for a singular difference. There's a couple different options that you could say for these ones. So I'll put down the two that I thought of. So passive transport, the most obvious one is that it requires no ATP. You can say it requires no ATP, it's spontaneous, no energy input, anything like that. And since it doesn't require any ATP, it is going with the concentration gradient. So it goes with concentration gradient. Or you could say it moves from high to low, which is with the concentration gradient. And then for active, it requires ATP or energy input. You can say it's non-spontaneous, endergonic, any of those. And then it goes against the concentration gradient. Or you could say it goes from low to high concentration. Any of those would have worked. They were only asking you for one of them. Okay, and then for B, it asks you to justify why the scientist used an equal number of red blood cells in each culture dish as a control. So they're really just asking you to justify why they used that control. So let's go back into the question. So scientists incubated an equal number of red blood cells in separate culture dishes that contained this 300 nanomolar solution of radioactively labeled glucose. So the reason that they would use the same number of red blood cells is because of what they're actually looking at as the experiment. So what they're really trying to look at is whether red blood cells lose the ability to take in glucose based on age. So you're looking at age versus ability to take in glucose. So if you were to plot this, you would look at, again, you don't need to plot it, but you would look at age here, and then you would look at like glucose uptake. So that's your independent, your dependent variable. And so a control that you would have to do is to make sure you're using the same amount of red blood cells in each trial. Because if you didn't, then you would have another variable at play and you wouldn't be able to see directly if age is what's causing that change. So since you want to look at age, so since your like independent or your changing variable is age, so since independent, or you can say the thing that you're looking at, independent variable is age. You have to keep all the other variables constant. So other variables like the number of red blood cells, you also need to keep the amount of glucose that you added as a control as well. So there's a bunch of different controls here. But basically, since you're only looking, like you're only attempting to look at the effective age, you can't change any other variables. Let's keep other variables as control so that you can like directly look at the effect. Okay, so that's it for A and B. Now for C and D, it says glucose transporters are required for facilitated diffusion of glucose into red blood cells. The scientists claim that the expression of gene encoding these transporters decreases as guinea pigs age. If the scientists' claim is supported by experimental data, predict the effect of increased age on the amount of radioactively labeled glucose present inside the cells of each group, and then justify your prediction. Okay, so you have your red blood cells and it's facilitated diffusion, which means that they just have to go through the transporters. Now, scientists claim that the expression of genes encoding these transporters decreases with age. This is the key. Okay, so if I'm decreasing expression, so decrease gene expression of transporters, you have to mention the fact that that would then decrease the number of transporters. So yes, it's a decrease in gene expression, but we know that a decrease in gene expression is going to result in a decrease in protein level. So you're going to decrease protein level or just decrease like number of transporters. As they age. So as the guinea pigs get older, since they are decreasing in gene expression, they're also decreasing in the number of transporters. So a guinea pig that is, 7 months old would have less transporters than one that is, 7 days old, for example. So they're asking you to predict the effect of increased age on the amount of radioactively labeled glucose present inside the cell. So you've got your red blood cells here, and then you've got your transporters, that would be, here. If it's an older guinea pig, it would have less, and if it was a younger guinea pig, it would have more. And it's saying that the glucose is being uptaken into the cell. So the glucose is going from here into here. Now, if there are less transporters, you would have a less glucose inside the cell. Just because there's less transporters, there's less things taking it in. So if you decrease gene expression of transporters, you decrease the number of transporters as they age. So you can say, therefore, older guinea pigs would have less glucose. In their red blood cells. Okay, so older guinea pigs would have less glucose within their red blood cells because they have less transporters. And the reason they have less transporters is because the gene expression of the transporters decreases with age. Okay, and that's actually it for number 3. So this one was pretty straightforward.